Now let's turn to segmentation, which is the process of classifying pixels as either foreground or background. Segmentation methods are used to partition an image into a set of non-overlapping regions whose union is the entire image. For microarrays, the non-overlapping subsets for microarrays are foreground and background. Probes are typically spotted on the array as circles. However, the spots may vary in shape and size due to variations in the printing process. Methods of segmentation include fixed circle, where all spots in the image are assumed to have the same diameter. Adaptive circle, which estimates the diameter for each spot separately. Adaptive shape, which estimates the boundary of the spot shape for each spot separately. Statistically based segmentation methods also exist and include ranked intensities and a method based on the Mann-Whitney U-test, also known as the Wilcoxon rank sum test. Here's an example of a spot patch, which is the full rectangular region where you can see somewhat of a circular spot within the spot patch and the individual pixels comprising this. There may be hybridization issues that affect the spot intensities, such as dust particles or contaminants that artificially contribute to signal intensity, or fragments of the probe may be inadvertently placed outside the circle yet counted as background. Some circles or spots may be smaller or larger than others. The spot could also take a different shape. When using intensity-based segmentation methods, we assume the signal pixels are brighter than the background pixels, as illustrated by these two densities. However, complete separation is rare, so statistical methods are used. Various intensity-dependent methods have been suggested, including rank-based intensity segmentation. Suppose each spot patch is stored as a 40 by 40 pixel image and that the spot diameter is 20 pixels. We know that the area of the spot is given by pi times r squared, where r is the radius. In this example, the area is 314.1593, while the area of the 40 by 40 spot patch is 1600. Therefore, approximately 20% of the pixels should represent foreground. The rank-based intensity method ranks all 1600 pixels in the spot patch, then declares those pixels in the top 20% as foreground and considers all other pixels as background. The Mann-Whitney U-test has been widely used for identifying background and foreground pixels. The spot or circle is segmented from the spot patch or square using a predefined mask, commonly a circle of fixed size. 8 pixels, labeled Y1 through Y8, outside the mask are randomly selected to represent background. The pixel values inside the mask are sorted in ascending order, and the smallest 8 pixels, represented by X1 through X8, are selected. Then you use the Mann-Whitney U-test to test the null hypothesis that the location of X is equal to the location of Y against the alternative hypothesis that the location of X is greater than the location of Y. If the null hypothesis is not rejected, n pixels from the observed X1 through X8 are eliminated and the next smallest 8 pixel values within the circle are selected. The hypothesis test is performed again. This procedure repeats until the hypothesis is rejected. A rejection of the null hypothesis leads one to believe that the observed pixels included in the hypothesis test are significantly higher than background. Thus, those 8 pixels and all other pixels within the mask with an intensity greater than or equal to X1 through X8 used in the hypothesis test are identified as foreground. Again, the purpose of segmentation is to partition pixels into one of two classes, foreground and background. Typically, background is identified so that a correction can be applied. Most often, background correction takes on the form of subtracting the background from the foreground signal estimate. Some issues with background subtraction is that it has been noted to increase the variability of genes, especially those with low expression levels. Notice if we let X represent foreground intensities and Y represent background intensities, from statistical theory, the variance of X minus Y is the variance of X plus the variance of Y minus 2 times the covariance of x and y. However, if x and y are independent, the covariance is 0, so clearly the variance associated with subtracting background from foreground would be expected to yield more variability. Finally, we will turn to intensity extraction, which is the process of calculating intensity measures for each spot or probe. After identifying the location, size, and shape of each probe and identifying background versus foreground pixels, gene expression values are calculated using some function of the observed pixel values within each segmented area. For example, the mean, median, total, etc. The method of data quantification used by Chen et al. In 1997 was to report the median of those pixels classified as foreground. When performing intensity extraction, the underlying principle used to guide selection of a method for data quantification is to select the statistical summary that best correlates with the amount of hybridized DNA target in the sample. Image analysis differs for Affymetrix gene chips. Because of the in situ synthesis, the perfect match and mismatch probes on the surface of a gene chip are rectangular and tiled next to one another without spacing. On the left, a part of a gene chip is displayed where you can see several rows that contain PM and MM probes. On the right, we have zoomed into one probe and can see the individual pixels comprising the probe. In fact, a given probe is a 6x6 grid of pixels. The outer pixels are removed, leaving a 4x4 matrix of pixels. The probe cell intensity is then taken to be the 75th percentile of these 16 interior pixels. Resulting data are saved to a cell intensity file with a CEL extension. Let's look at an example. These are actual pixel level intensities from a gene chip for a specific probe. Within the probe, there is a 6x6 grid of pixels for which we have the intensity values. Note the intensities around the perimeter are lower. Eliminating the perimeter pixels or outer edge, we are left with a 4x4 matrix of foreground pixel intensities. The 75th percentile of the interior pixels is taken to be the probe level expression value, which is 594.